In eukaryotes, DNA transcription begins when general transcription factors bind to the promoter region of the gene.
So that, for example, you see what I was talking about at the end of last class. That you have the part of the gene that's the actual transcription code or unit that has the information that you want. And then you have right upstream from that a promoter box. We kind of talked about that. And then you need these transcription factors in order to activate a transcription, because RNA polymerase is unable to attach to the DNA to transcribe without the transcription factors in there. So that we're going to talk about today about cell communication, right? And then we're going to talk about that these transcription factors are going to be the what's going to generate the response. Cell communication happens in 3 stages: reception, transduction, and response. So you can think of the transcription factors as the response. If transcription factors for a specific gene are present in the cell, then the gene gets transcribed. If not, it doesn't get transcribed. Last class we talked about epigenetic control of gene expression, and how you are what you are because of the environment you are in. You are what you are because of the DNA that you have. But the DNA that you have also only responds depending on the chemical environment which is in. And this is what I'm talking about. So without the transcription factors there, even if you have the DNA there, the gene doesn't get expressed. Continuing, it's more than that.
Transcription factors facilitate the binding of the RNA polymerase enzyme to the start position of the gene. RNA polymerase.
Just remember that a common box, a promoter box, is one that repeats a lot of A's and T's. It's called a Tata box in humans.
Needs to be activated by a type of transcription factor called activator.
Now, remember, if you're talking about bacteria, you sometimes have in the beginning of the gene something that either blocks the gene from being read or something that turns on the reading of the gene. So something that would turn on the reading of the gene, like it is on the lactose gene, the gene is on or when the lactose is present, but only if there's not too much glucose already around. So that's like the positive control thing. So in the case of positive control, you need an activator to start. And I mentioned to you guys how in eukaryotes, most of the genes are activated. So like most of it would be, if you put it in terms analogous with that, what's happening inside the bacteria, it would be kind of a positive control. So a special type of transcription factor called activator is usually the last one to kind of combine here and turn this whole thing on. Now remember, all of these things, well, RNA polymerase is a protein, transcription factors are proteins. So I kind of ended the class talking about this last time, that even if you think about the fact that this is a protein, therefore coded by a gene. No, sir. Get set, sit down. All right? First 15 minutes, the last 15 is a class. All right? So if you remember that this is something that gets coded for and transcribed like everything else, that in itself, there's another gene somewhere else in the genome that needs to be on in order for this to be here. So you can think of him as like that whole epistasis thing. This gene that's going to code for a protein or an RNA type will only turn on if the genes that turn these proteins turn on. And in that way, the DNA of humans is, or any animal really, or any life form really, is organized, that it's controlled that way. Genes turn on other genes because sometimes the proteins that a gene codes for serves as a transcription factor to turn on another gene. Is that clear? All right? So you can think of that as a distal control mechanism. Somewhere else in the genome, you have the genes that code for the proteins, which then activate this gene. So genes depend on each other, so to speak.
Some activators recognize and bind to the regulatory region of DNA near the promoter region. Other activators bind to a distant region of the chromosome called the enhancer region. Activators bound to the enhancer region work with mediator molecules to...
I kind of explained this last class. I wanted to show you. That's why I'm showing you the video. That sometimes the control mechanism that turns on the gene is actually in a completely different segment of the chromosome, and sometimes in an entirely different chromosome altogether. And so you need the activator to activate that gene, and then for that to serve You see there's another protein here, acts the mediator. Now, so all of these proteins that are doing these jobs, right, have to be around in order for the gene to be expressed. So therefore, it's why it's beneficial for a gene that wants to be expressed, or if the gene needs to be expressed a lot, for that gene to be in the area of the cell where a lot of these proteins are present. Because these activators, or these mediators, molecules, or RNA polymerase, will be present in greater amounts in the areas of the nucleus where transcription is happening a lot. Now these orange ones here, the transcription factors, are specific to the gene that needs to be read. In other words, they work only, typically, in that gene. And the funny part is that a set of complement of transcription factors is usually necessary. If you go back to the beginning, it had 3 or 4 different proteins combined together to create this initiation complex. So that means that you would need all of those 4 things. Yes, it's like a little checklist. I'm going to make the gene if this, and this is happening inside the cell. Each transcription factor is a message. When all 4 messages are present, then you start the process of doing that. So you can take, for example, like we talked about cell cycle control. So when the cyclins and the CDKs combine, and you have them one activating the other, what are they really going to do? They're going to activate other molecules which will serve as transcription factors that will activate a series of genes involved in initiating the replication, probably making a lot of DNA polymerase to make sure that the DNA is copied. Likewise, any effect that you want the cell to do has to start with the transcription factors being present. So the DNA is only read if the proteins that turn it on are there, which is kind of like the kitchen. So like, in other words, DNA is being read to make proteins, but it only gets read with the help of a protein if all the proteins are already around. And all these mediators and activators are not necessarily specific. They are just there to activate any gene that needs to be turned on. That's why you want to be in the area where all of them are around, so that you will be activated if you need to be activated. Yes?
Was this what you meant when you drew it on the board?
Yeah, that they kind of fold inwards.
Oh, okay.
Initiate transcription. Another type of transcription factor, called repressor, inhibits the activation of transcription. Repressors can act in different ways. Some repressors bind to the binding site of activators and prevent activators from binding to DNA. Some interfere with the molecular interaction between the activators and the RNA polymerase.
So eukaryotic genome 2 can actually have these repression mechanisms, but it's not exactly the same as the bacteria one we saw that kind of just blocks the promoter box, right? I mean, remember, there's 2 types of those. There's the one that bystander is blocked and you have to turn it off by removing it, or the one that bystander is open and then the repressor goes there if it turns on and it stops it.
Some activators and repressors act by affecting chromatin structure. These repressors can alter the chromatin structure to make it coil up tightly. Coiled up genes are not available for transcription. Conversely, activators can uncoil a coiled section of DNA and make genes in those regions available for transcription. The interactions of all these factors determine the actual rate of DNA transcription.
So it's much more complicated, right? Because you have to take all these things into consideration. So when you're trying to decide whether or not a gene gets expressed, it depends on a whole facet of chemical interactions that occur at the nucleus to control that. All right? So it's very complicated, and that's why with only just a few genes, we can have a great degree of variety among eukaryotes because of all of these different layers of control. So then, to kind of we're talking about eukaryotic gene activation. So then, this kind of brings together the second method, because the first method we talked about was the idea of structural control, which they kind of mentioned there, the whole binding versus coiling versus uncoiling. So an activator could, for example, acetylate the histones, which make them open up, or a repressor could methylate the histones or the DNA strand itself to make it coil up and be unreadable. But these things we talked about today were the whole regulation, the chemical regulation of initiation of transcription. There is another method of control, which is, so again, by the way, these enhancers and activators, control elements are all, remember, all proteins. And that's very important because you need to have the genes for those things on in order for the genes that you need to on to be on. Right? And then today we're going to talk about cell communication, and after that we're going to talk about how cells become determined. And then all of this has to be in the back of your mind when you're thinking about that. So then, okay, so enhancers and activators, we already talked about this. Let me see. See if I have anything here that I have not mentioned. Oh, yes. Okay. So, let me just put this bigger. These are just key terms that remind me to say things that I wanted to say. So, did you notice that the activators are proteins, right? But they interact with DNA. So this is going to be an important thing we talk about biotechnology in the coming weeks. If there are pieces of protein, right? But because of their structure, their domains, the shape that they have, which are attracted to specific sequences of DNA. So this is going to be important. Say, for example, if you want to tag DNA. Let's say you want to do some sort of research that you want to identify a specific gene, right? Well, you can do something similar to what these proteins do. You can find the gene sequence that you're interested in, and then have a protein designed to kind of hybridize with that, is what the word is. So if that protein, though, is tagged, say, for example, with the radioactive chemicals, then the part of the DNA where the protein is would light up. And that's one way for you to find in the chromosomes where the gene you want is. If you think about what I'm saying, there's probably a video like this that just does a better job at visualizing it, but using your imagination can be better. So if you have a sequence of DNA that has a specific sequence, whatever the sequence is, and you're looking for that specific sequence, there might be a protein sequence that will recognize that sequence. Now, if this is part of a larger protein that's radioactively tagged, when that protein binds with the DNA that it wants to bind to, it's kind of like color coding the DNA. Does that make sense? Mm-hmm. And with that, you can do what's called a cytogenic mapping, which you can find where in the gene, in the genome, or what chromosome each gene of interest is, right? So that's kind of like one way that we. This is called hybridization, and that's a very important way to detect DNA that you want. You can do the same thing to detect RNA. So let's say, for example, you are trying to do a study to find out whether or not certain types of RNA are in the cell at that moment. Why would you want to know that? What would it mean if the RNA is present at the cell at that moment? What does that mean? When RNA is around, for one specific RNA is around, why would it be around? Because the gene is being what? It's being expressed, correct? It's going to be translated. So the presence of RNA indicates the presence of the cells trying to express a protein. So by examining which RNAs are in the cell, you can learn about what the cell's trying to do, if you think about it that way. So we also do use techniques like this of tagging the genome to try to find what parts of the cell are. So for example, they actually call this northern blotting, the one that has to do with RNA, and the one that has to do with solid blotting, the one that has to do with DNA. So what you do is you get a little vial full of these little wells like this, and in each well you put one sample of RNA, right? And then you cool the entire well with a certain protein that's trying to identify one specific sequence, right? So like maybe that well came from the cell at minute 4, and then that one cell from minute 8, minute 12, minute 16, minute 20, and so forth. And in that way, you create a timeline of the chemistry inside the cytoplasm of the cells. Are you with me? And then you use your priming technique where you're trying to hybridize the RNA that's there with a protein that's tagged with a chemical, right? And then you literally put that over that, right? Of course, the cells, the wells that have the RNA that is the one I'm looking for will be tagged and will start becoming colored, right? And in that way, you identify the presence at which cells had the RNA you were looking at. So you can do that to compare different tissues, for example. So if you try to compare several different tissues of the body for the presence of a certain type of RNA. So you're trying to find So you get in the body, you're doing several different tissues and try to find is the RNA present in this type of cell. So you compare a muscle cell. So every one of those vials is a different type of cell. Or you can do the same technique to say figure out when doing the development, does a certain gene turn on? Think about it that way. So you get the cell, this is day 1, day 2, day 3, day 4, day 5, day 6, and then you can actually see at which point the gene turns on. How you know the gene is on? Because the RNA is around. Does that make sense? So that means this can let you figure out the process of determination of a cell if you little by little, step by step, minute by minute, detect for the presence of all known RNA types that show up during development. You can actually figure out the sequence of sequential activation of genes. Does that make sense? There's people working on that. Literally, little by little, one RNA at a time. You know, minute by minute of the development thing, trying to figure out. Of course, we're doing this in things like worms and rats, and probably in human embryos too. But it is a tremendous undertaking. There's thousands of genes, right? Thousands of human genes. And each of those genes can be coded differently depending on which introns you leave or do not leave, right? And so for you to scan each cell for each type of RNA that we know should should be around during that time, right? And then remember that you want to know exactly the order. So what if you're 1 second early or 1 second late? And then you have to repeat it multiple times because you want to make sure it's a pattern and not an accident that happened in that cell that you're looking at. So this is something that's going to take a very long time. But when we can study that? Remember, this is called northern blotting. So I just explained what northern blotting is. Southern blotting is the same mechanism, except you're doing DNA analysis instead of RNA analysis, right? And it literally looks like that. They put the samples in the vials of multiple different sources, and then they literally wash. It's called a wash the vials with the hybridization factor, and then you put it with pressure on top of the thing. Let me show you a picture of it. So, where is it? Because one of the things that we did not do as 3A is talk about some of these techniques that we had to talk about. So there it is. It's kind of like exactly what I just said. So you put, well, I've skipped a step that we didn't talk about yet, but first you need to separate the DNA or the RNA that you want to. You have to extract it from the cell. So first is extraction. Extraction is just a mechanism of separating the DNA from the rest of the cell. Then you have to separate it by size so that you can identify the gene you want. And electrophoresis does that. We'll talk more about that another time. And then that's once it's separated by size, you can just do what the blotting is, which is you're going to put it on a vial that has the wash, and then the ones that are the ones you were looking for, like become hybridized by the probes. Probe is the name for this protein that it will attach to the genome you're interested in. And so let me see if there's a better one. There's another example. Like you literally have the resultant gel from electrophoresis, and then you put that into in this method, they actually put in a bag that has the wash, like the chemical that has all the probe in that, right? And so the sequence that has the DNA of interest becomes tagged, and then you know that that was the one you were looking for. So it's kind of a lot of. Let me see if I can find one that looks like the result one. So when you do electrophoresis and you get a result of electrophoresis, it will look something like that. But then you can wash it with the probe, and then the one that has the code you wanted becomes colorful. Does that make sense? It will get the same color as a probe had. Does that make sense? So you get the gel that comes out of electrophoresis, and then you wash it with the probe, right? And then whatever one. This northern blotting has to do with finding RNA, and southern blotting has to do with finding DNA. But it's the same idea. So they have here another example. Let's talk about electrophoresis since we brought it up. Okay. Now this is part of Lab 8, pre-lab. It's the idea that you have to kind of work on that, but it's a chamber, and then we're actually going to be working on this lab, provided they give us the materials, in kind of teams, because I want every single one of you guys to have the experience of creating the electrophoresis, and we only have 2 chambers, and so we have to take in turns. You know what I'm saying? So, but either way, you hook it up to a power supply. I don't know if you did this when you were in chemistry, but if you run a current through an electrolyte solution, right? So that water, even if it's just water, you get these bubbles forming because you basically separate water into hydrogen gas and oxygen gas. One of the probes, one of the sides makes oxygen. Well, it's actually easy to figure out. The positive side and the negative probe. Like, so let's say you have an anode and a cathode, and all of that is inside of a liquid, right? That has, well, let's say if you just do it with water, it works too. Well, oxygen, remember that water kind of, it is H2O, but it actually also separates itself into OH, and right? And as long as you don't have an acid or a base, you're going to have the same amounts of H plus and OH. The H plus actually reacts with water to form something called a hydronium ion, and that's the actual ion that acids have, right? But either way, as long as you have pure water, you have the same amounts of each. But you see how this is kind of like the negative end, so that this gets attracted to the cathode,
Please
right?
this interruption.
And then
For
this
all
one
students going
gets.
on the Martin's field trip, please report to the
Anybody else going
cafeteria.
on a field trip now?
All students going on the Martin's field trip, please report
So
to
if
the
you're going, go to the back
cafeteria
door,
at
please.
this time.
All right, this is being recorded. Hopefully you can. But anyways, the oxygen gas will form in one of them, and the hydrogen gas will form in the other one, right? So it'll be attracted. Are you with me? I'm saying, and then you're gonna get these bubbles. You're gonna see the same bubbles. If the electrophoresis chamber is working, you see the same bubbles. But the point is, for this to be true, usually they put there's a current running through it, and that means that needs to be in a kind of like an electrolyte gel. It has to be have cations or anions in there. So we have this gel, which is like it's a prepared gel, and not this gel, this liquid, the electrolyte solution, and then it's kind of like a bath, and in that bath you put a little gel. That gel, it's like literally made of like the same kind of thing that the agar, that we make bacteria grow, which we're going to do in lab 8, right? And then, but it has these little tiny holes on it, and then in these tiny holes, which is the harder part of the lab, you're supposed to like put the DNA sample in there. Now we have different types of DNA sample, and because the DNA, okay, pause this, let's talk about another thing, which is restriction enzymes. Now, you know I told you that there's proteins that have domains designed to recognize specific sequences, right? Now, in the case of a probe that you're using for northern blotting or western blotting, all you wanted to do is really attach to it so you can kind of color code it, right? But a restriction enzyme does another trick. It recognizes a specific sequence and then cuts right there. So that if you look at what a restriction enzyme does, is this. For example, let's say there is a specific sequence of DNA. In this case, is that thing right there. Then there's a restriction enzyme called E-Cori, which is the one we're actually using in the lab, that cuts it. And it breaks it into these sticky ends thing. It's called sticky ends because it didn't cut it like that, right? It cut it leaving the sticky ends, which is nicer if you're trying to do genetic combination because it makes it easier for you to insert that sequence into another DNA. I'll talk about that in a second. If you're doing, but basically the idea of a restriction enzyme, is an example, is that it will cut the DNA at a specific sequence, leaving that. Now, some restriction enzymes do leave sticky ends. Others just cut it, like just get the double helix and cut it like that. Are you with me? But whatever it is, each restriction enzyme recognizes a different sequence. It's kind of like the spliceosome, right? Each spliceosome recognizes the point to call it an intron and cuts that out. So a restriction enzyme, it's the same idea. It recognizes a sequence and cut it. Now, if you use a different restriction enzyme, you're going to cut it in different ways. So you think of it as a scissor that only cuts at a different place, right? So if you use the blue scissor, you're going to create 20 cuts. But if you use the green scissor, you create 2 cuts. Or if you use the yellow scissor, you create 50 cuts. Are you with me? So each type of restriction enzyme will cut the DNA in different chunks of different sizes. Does that make sense? Right? Because you want to do something with that, right? So it's about cutting the DNA into chunks. Why? I'll give an example now. So, but clear, first of all, is the idea that each restriction enzyme is going to cut the DNA in different chunks. So here's an example of why you wanted to cut it, right? So you get a piece of the gene, you want to put that, you want to insert that, that inside of a plasmid in order to give a bacteria some new feature that piece of DNA is going to give. Let's say it's a glow-in-the-dark kind of feature, right? So then what you need to do is you have the restriction enzyme there. It's designed to cut it noticeably right after the promoter. Why would you want to do that? So then you can put the DNA right after a part that's going to tell it to express that. Does that make sense? So if you have a promoter that's usually on a lot, that's a great place to put a gene, because that means that it's going to get expressed a lot. Does that make sense? So if you find a bacterial DNA that's commonly on a lot, that's a perfect place to put a gene there, so it gets expressed a lot, right? So anyways, you get the restriction enzyme to go there and then basically cut it up. And then once it's open, you can then insert, because it has that sticky end. Are you with me? You can make sure that the DNA you're trying to insert has a complementary sticky ends. Are you with me? So you left a sticky end on one side and on the other. You see? And then you make sure the gene you insert has a complementary sticky ends. Right? And therefore it will already have the affinity to get into it right there. And then you insert it, and this is how we do it. And now let's say you want to make the yeast produce insulin for you, and that's how we do it. We insert the gene into the thing and then put that plasmid inside the yeast, right? And now the yeast starts making it has the gene to do something they didn't have before. And we use this kind of thing to all kinds of applications, to give plants faster growth, or give plants the ability to produce a toxin to defend from a pest, or to give the plant the ability to resist, to grow with less water. So we find something in a plant that we like, and we say it would be nice if our corn had that. Right? Oh, look at this plant. It barely needs water to grow. We have a problem with drought right now in California. No problem. Let's put that feature of that plant inside of our corn so that we can grow the corn in a drought. Look, are you with me? And so it's you give the ability. Ever seen Deadpool? Right? If you watched Deadpool. So the idea, what they do in Deadpool, is that they actually give them the mutant gene, the X gene, right? Which is the gene that the mutants have. So he's not a mutant, but they insert the mutation in him. In effect, what they did is some sort of gene therapy with him, right? But all the cells of his body, though, have been mutated. We're not far from being able to do something like that. But we do that stuff with embryos, though, all the time. So let's say if you get a rice embryo, then you just want the rice embryo to be a better rice embryo. This is called Gmo food, right? Genetically modified organism. We do it with cows, we do it with chickens, we do all kinds of stuff. Now, some people say that some of these GMOs, when fed to cows and other cause diseases in them. I mean, some of them, not all of them. You know what I'm trying to say? But people say that there's bad things from GMO food. I'll make a little argument here, which, by the way, the science is still out. There's no definitive evidence on whether or not all the GMOs or some GMOs are bad for you or not. But I will make this argument. There is no such thing as natural food. All right? All the food that we eat is been artificially selected for. Correct? If you think about it, the corn we eat today, or the rice we eat today, or the wheat we eat today, the cows, the pigs, everything, over thousands of years of human agricultural enterprise, we've selected for features that we wanted. Correct? Mm-hmm. What all the GMOs do is they let you skip natural selection and go straight to the look you want. Think about it. If you're trying to select the plant that can grow in the driest conditions, you have to wait several generations for evolution to do it. And you saw how hard it is to grow 1 or 2 generations, which we're still waiting also for the materials to keep doing with that. But imagine going 5 generations, 10, 50, 1,000 generations to finally get the look you want on 100% of the generation. Are you with me? Or you just skip all of that and put the gene on there. That's all it is. That's all GMO food is. It's basically skipping artificial selection and putting already what you want in the genome. Does that make sense? So if you're going to argue against GMO, you might as well argue against all the food we eat. Does that make sense? Because we've already done that kind of stuff over generations with all the food that we eat. You know? So I don't think the logic makes sense. You know what I'm trying to say? Like, in effect, what we're doing now is being able to modify food quicker. And it allows us to, we talked about this already. It's a new form of evolution. Does that make sense? We can make bacteria and yeast make proteins because we do not have artificial ribosomes, a protein factor. If you are a biochemist that design a new protein to serve as a medical intervention, so I'm trying to fight cancer or fight something, right? And I made a new protein molecular structure, and I designed a code for it. So I reverse engineer a protein. What does that entail? It entails creating the shape I want, figuring out what amino acids I need to put in sequence to create that shape. So, and then reverse engineering, or going backwards translation, figure out the RNA code I need, and then reverse transcribe to figure out the DNA code I need. Now I have the DNA code I need to make a protein that doesn't exist yet. This is called, molecular engineering, right? And all of it so that I can make a better drug. Does that make sense? Or a drug that works for that specific type of cancer, or that specific medication. So when antibiotics running out, that could be the promise of the future. Engineer the molecule that kills the bacteria instead of waiting to find one that does it. Does that make sense? So we need to engineer the molecule. However, we need to build it. And then the only thing we have to build it is ribosomes, and we can't make them. We have to use the ones that exist. Ribosomes are an incredibly complex molecule. So what we do, we put in a bacteria that already have the ribosomes, and then we gotta give them the DNA. So this is genetic engineering. I hate the idea of doing that. And this is why, one of the reasons we use restriction enzymes. The other reason is for DNA analysis. Now, following DNA extraction, okay, so now in extraction, by the way, I'm doing a review of biotechnology here too, okay? So lots of cool things. I didn't mean to do this today, but somehow I got there. So really what you do is you get the cell, so several cells. Let's say you're found in a crime scene, a little bit of, or you doing the rape exam, you got found some semen. So you get the semen specimen, and you need a lot of it now, correct? But the first thing you need to do is to get the actual DNA that's there. So the way it works is you put that specimen inside a chemical regimen that is designed to break down the cell membranes. So the first thing you're going to do is something that's going to dissolve the cell membranes. When we do strawberry DNA extraction, I always do it for all biology classes, even though it's something that people do in middle school, because it's kind of fun. And except that when I do it, I tell you guys can do whatever you want. You can bring whatever you want to do with it. If you spit enough in a cup and bring that, you should be able to do it with your DNA. You know what I'm saying? If you bring a apple, if you bring a whatever you want, you can do it with. As long as it can become mushy, right? You got to make it mushy. So now, of course, in a real laboratory technique, they add a chemical. There's a surfactant, most likely, to destroy the membrane. Then they put it in a centrifuge, right? And the centrifuge will spin it. And when you spin it, by the way, they also they usually put a solvent that is designed to separate things by density. In our case, we put like a bit of alcohol, and then it usually separates the DNA, makes the DNA float to the top. It doesn't work all the time though, but they will find the right solvent to separate the cell parts by density. And then, the way that Avery did it, when he was trying to redo the experiment of the rat to figure out which part was actually doing the killing, right? Because Avery redid the experiment that Griffin did, when he was trying to figure out something about the deadly strain was going inside the good strain and making the deadly strain, the good strain to deadly strain, right? So, but Avery redid that. Well, he used a solvent that made the DNA sink, and then that's a classic picture that shows. If you look up Avery's experiment, the image. You gotta get some other girl named Avery.
McCarthy
this is McLeod.
McCarthy and McLeod. It's McCarthy, Avery McCarthy and McLeod.
Let's do experiment. Avery experiment. So yeah, what he did is he separated the parts of the cell into different parts, and then examined only the DNA. When he did it with the DNA or with the chromosomes, then it worked. So that's extraction. So you put it in the centrifuge because the centrifuge will spin, and then things which are denser will separate differently with things which are less dense. So in addition to the solvent, that will help separate the cell parts in different parts. And then when you get all the DNA, you get a little pipette, you grab the DNA out, and then you do what you need to do with the DNA. Now, that's extraction, right? Culture. Culture is just getting a cell that you're going to do it, right? You're going to do it here, and then this is lab A. You get a cell culture that's growing, and you transfer it to another place, so it keeps growing, and then when it consumes all the food, you're going to get some and transfer to another place, and so forth. So culturing is just kind of like keep a cell line alive. Does that make sense? By keep transferring to new places where they can grow. So you get one cell. Let's say, for example, you swab the doorknob and you put it in the agar. The cells will start growing on it, but then you're going to find out what is that. Okay, I want to keep it going because I want to research this for a very long time. So one of the things you need to do in the background of all your research is keep growing them in more and more plates, because eventually they die, they consume all the food that's there. So what you got to do is you got to once all depends on your regimen, but this is one of the things I did when I was in the lab. Almost every week we have to make new cultures from all the things we were working on. So if you get the cancer tumor of the patient you're examining, right, like Henrietta-Lacks cells, you're gonna get the cells, make them grow in a culture that has all the food they need, right, and then every week you get some of those cells and start another culture, so that they live forever. You know what I'm trying to say? It's kind of the idea, so you can keep studying that thing that you're looking at. That's culturing. Extraction has to do with getting some sort of biological sample, using a surfactant to get rid of the cell membrane, so now you have all the parts of the cell just in the solution, using some sort of solvent to help separate by density, and then putting in a centrifuge to do that more accurately, and then you can get the part that you want. You get the DNA out, now you can do whatever you want with it. One of the first things we need to do with the DNA is copy it. Because if you want to study the DNA and have a lot of evidence, you're going to need multiple copies of it. So there's two mechanisms to copy it. So let's talk about cloning first. Now there's two types of cloning. The cloning that we talked about already in this class is the cloning of a living organism to make another copy of the whole organism. It's different from what is referred to as DNA cloning. So DNA cloning is this. Making multiple copies of DNA. And the way it usually works is that you put that DNA that you want a copy of inside of a plasmid, and then you put that plasmid inside of a bacteria, and then just wait for the bacteria to make millions of generations, which only takes a few weeks, right? And then when you come back, you have 1,000,000 times the same amount of DNA. Does that make sense? So use the cell's own machinery of cell division. So you basically insert the plasmid inside of a bacteria and put the bacteria in a culture, and you just keep culturing for a few weeks, and all of a sudden you have from one cell that has, or a few cells that were modified, you now have thousands. Does that make sense? But you still have to wait 2 weeks to get 1,000,000 copies. So then that's what we used to do it, and sometimes we still do it if you want to have what is called a live bank. A live bank is a place where you have the gene in a living culture. Does that make sense? So Eukaryotic cells is an example of a live bank. We don't need to have live banks anymore because we can have a data bank, the genetic code in a computer, and we can actually produce the genetic code or print it biologically by literally creating the code with a machine, which we'll talk about in a second right now, writing the DNA code, and then inserting the bacteria if you want to. In other words, you can print a molecule of DNA and then put it inside a bacteria, so there's no longer a need to keep a live bank. Does that make sense? Once you have the code, you put it in the computer, and then whenever you want the live bank, you just print the DNA. I'll explain how that works in a second. And then put it inside the bacteria. Does that make sense? So if you ever need the living specimen, you can just make it from the computer code. With that in mind, think about how cool this is. That means if I have a virus in Asia, I no longer have to put it in a tiny container and carefully transport an airplane to CDC in Atlanta to study it. I just take a portable DNA sequencing device, right? That probably nowadays is as big as this book. And on the site, I sequence the virus, right? I get the code, upload it to the internet. Someone in Atlanta downloads it and prints the virus. Does that make sense? So I don't need to, so you can transfer life. So let's say, for example, we send somebody to Mars someday, and we want to send them a new type of plant, right? We don't need him to send a plant to them. We're just sending the code of the plant, and he prints the plant himself. Does that make sense? So that is actually possible today, and the same kind of guy that was in charge, that was, we're going to talk about in a second, that invented shotgun sequencing, which is a method of quickly sequencing the DNA, right? He also is the one that's working on that. Actually, his company is working on 3 major things. First one is this whole idea of DNA databasing, which is getting all of life in a computer, right? So that they actually have this, they go around the world collecting samples and sequencing everything they can get their hands on. The second thing is this, making this whole DNA printing thing quicker. Because sequencing is very quick, but the printing is still kind of slow. So making that better, eventually we'll be able to do that. And the third thing is actual synthetic life, which is the ability to create life from scratch, including all the ribosomes, the cell membranes, everything. All right? But this kind of, it's Prague's inventor, look him up. He's like very, 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 it doesn't work for any university. He works by himself. But anyways, are we clear on cell culturing? Are we clear on extraction? You should know what those things mean, right? And now you should also know what it entails. What do they do to do it? And we also know what DNA cloning is using bacteria. But we have a better way of DNA cloning. It's called PCR. So for that one, I'm actually going to show a little video. PCR. This is called polymerase chain reaction. And sequencing genomes would be impossible without it. So when you want to do sequencing, is instead of having one copy, if you have thousands of copies, it makes it a lot easier. So that's actually the principle behind shotgun sequencing. You're going to get thousands of copies, and you're going to copy, you're going to sequence. It's like this. Instead of sequencing, you know how the cell copies itself when it's making DNA polymerase? We learn it doesn't copy one place, and bacteria does it like this. It starts one place, and it goes around, right? And it just copies like, actually there's two orders of replication, one replication for it, and it goes around the whole thing, right? Now, we eukaryotes make multiple bubbles in multiple chromosomes all at the same time, right? So, Prague said, well, that makes sense. Instead of copying the sequence one by one of an entire enormous chromosome, what we do is we cut the chromosome into tiny little pieces, and we sequence piece by piece, and then we just piece the whole thing together afterwards. Does that make sense? Now, you may think, like, how the hell does he do that? So, because if you get this enormous piece of DNA, and you cut it into a tiny bits of pieces with restriction enzymes, again, another use for restriction enzymes, right? How do you know how to piece it again? Well, because you don't cut it with just one restriction enzyme. You also cut it the same copy with a different one, right? So then when these get sequenced, you don't really know where to piece them together because you're going to separate them all. So then at the end of the day, you just have a jumble of tiny little pieces. Multiple copies of tiny little pieces. But then these pieces, you see how it contains that piece plus these sides? Are you with me? And then that piece contains part of these two pieces, right? And you do the same thing with another restriction enzyme and another restriction enzyme. So you do that, and you use computers to kind of do the jigsaw for you. Does that make sense? Because while this piece here, by itself, you couldn't piece it together. Well, with this and this, you can piece them together. Are you with me? Because think about it. This piece right here will tell you how to put these two pieces together. Does that make sense? And then that piece right here can tell you how to put these three pieces together right here. Does that make sense? Mm-hmm. Now, there's no human doing this, a computer doing it, right? And it's not doing just two restriction enzymes, you do 50 different types. But then you cut the chromosomes into lots of different ways to make all these tiny little fragments, and then you piece them all together. By the way, I think that's formidable evidence of the power of computational technology to do something that people think it's impossible, which is to get fragments and build a whole lot of it. So, you know, people say, we don't have enough, the fossil record is all fragmented. How the hell could you tell the history of life from the fossil record? Well, how the hell could you piece together all your genes? All right, it's called science, bitches, right? So, now, before you can actually do sequencing, which we'll talk about in a second, we have to copy it so that you can get each copy and cut it differently and do this kind of thing. So, let's talk about how that works. Polymerase chain reaction. I'm sure you guessed this already, no, right? So these are the things you're going to need. You're going to need your original sample from which you can extract the DNA. We already talked about that. A PCR machine. And then multiple copies. So we find a specimen in Spain and want to do multiple copies. It's just a machine. Alright? Now, what the machine actually entails is a thermocycler. Alright? A thermocycler is going to be a machine that repeats heating and cooling many times. In between, certain things are happening. Primers, anybody knows what primers has to do with DNA replication? Remember this? DNA polymerase? DNA polymerase only works if it knows where to start copying from. So you got to have these little RNA primers to start. Now, of course, if I want to copy this and that alone, that's the only piece that I want. I want my primers to have the hybridization sequences. I talked about hybridization sequences today, which are they have a domain designed to identify a specific DNA sequence. So I want to make sure that their primer sequence matches the beginning and the end. Does that make sense? So I can mark where to start copying from, right? Because I don't want the rest. I only want my target. Because that's the part I want to analyze. That has the sequence that I'm interested in. Does that make sense? So let's see how that works. Of course, you're going to make DNA copies. So you need a lot of nucleotides sitting there. Just like the nucleus, right before division, has to have all of these nucleotides there for you to be able to do it. You also have to have polymerase. Now, the interesting thing about this polymerase is that you're going to put it in a heat cycle. That means it can't be any polymerase. It has to be polymerase that is okay being overheated. Now remember, polymerase is a protein. What happens to proteins if you overheat them, typically? They denature. So they had to find polymerase, a naturally occurring polymerase that wouldn't do that. So where do you think they got them from? Bacteria that lives in volcanoes. Very good, right? These archaeobacteria that live in extreme environments. So they got the polymerase from them, and then that's the polymerase they use. And let's see what they do. They put all of that in the vial. So they basically have the thing that gets the copy, the raw materials to do the copies, the primers, which will tell what part of the sequence I want to copy, and the original piece of DNA. The rest just happens by itself. Let's see what happens. So you put that in the thermocycler, and you can do multiple at once. Most PCR machines let you do multiple vials at the same time, right? And then you're going to turn it on, but what's happening inside of it is that the primers will then recognize that sequence. Right? The target piece of DNA will be primed at the beginning and at the end with the primers. So, but to do that, I need to denature the DNA. Just like proteins, DNA is held together by hydrogen bonds. So to denature it, all I have to do is heat it up. So you heat up the whole thing to 95 degrees. What that does is it denatures, just like the helicase would do. It opens up the strands. Now the primers come in, right, to identify the starting spots, right? Because RNA polymerase is like, where do I start? Where do I do this? By the way, it can work on high temperature because it is one of those NOSEP things. So they're looking for a place to start, but you need the primers to tell them where to start copying from. You design primers that match the initial sequences that you want for the target things, all right? Now you cool it off so that the primers can attach themselves. You actually design the primers to do that. Once with the primers in place, RNA polymerase will then attach itself to the beginning and start copying. And that happens at 72 degrees Celsius, which is when this RNA polymerase likes to do its work. Now notice what I just did. I made a copy of my target DNA. It's here. Of course, these primer parts have to be replaced with RNA primers, but that gets done by RNA polymerase as well. But either way, I am almost complete with my target DNA. But now notice I have two new copies of my target DNA. So that's not much, but at least it was faster than it would have been done by waiting for the cell to cycle in the bacteria. Now notice that I have these two original pieces with the primers there, and I don't really want them. I want the entire target DNA. But let's see what happens if I go another cycle. So if you keep going another cycle, the same process will happen again, and then you're going to get more copies, right? Except this time, when you get copies, and they'll show you, right? It will start from the primers again, right? So some copies. So see, this copy is now prime. You just keep watching. You see the piece you want becomes more and more common as you go through more and more cycles. So now you go to 3 cycles. Now see, at cycle 3, something magical happens. It's the first time you have a target molecule by itself. Does that make sense? Because until then, by the whole semi-conservative process, you have half old, half new, right? But by now, you start getting these targets by itself, because you notice that since I'm only copying from there to there, right? It just kind of, if you go back one step over here, RNA polymerase doesn't know where to stop. So when it copied an entire new molecule. You follow what I'm saying? So it just went through the whole thing, and that sucked. It wasted effort there. But since this is a beginning, and this was a beginning here, wow, if I separate it again, that part gets separated by itself. So as I do more and more cycles, I have less and less of these entire copies, and more of the piece of only the pieces I want. Check that out. So I'll keep doing it one more time. So see, now after this cycle, I'll have more of the target fragments and less of the interior pieces. You see it? And then, so if let's say I do millions of cycles, right? Eventually, I'll have so many of these to these that I barely have, statistically, I have none of them. Are you with me what I'm saying? So look at that. They're going to show you now, they're going to accelerate the process and show you how it works mathematically. So after the third cycle, you have 2 copies, complete copies. At 4 you have 8. And then as you keep going more and more and doing more of these copies, so the cycle, this is why it's a polymerase chain reaction, because as you do it multiple times, it gets exponentially bigger. By the time you get to cycle 18, you basically have none of the other one, right? It's like you can't even find it. But a typical PCR will cycle in 6 hours or so. I used to do it all the time, which lets you go about 50 cycles. I think they don't get that far. But when I used to run PCRs, you do about 6 hours, and you get literally like half a billion copies of the DNA. You know, so it's exponentially bigger. So now that's much better than cloning, because you don't have to wait for the cells to copy themselves. And also, when you do cloning, it also skips the. When you do PCR, it also skips the extra step of having to extract it again. You know, because you have to extract the pieces again. And so this is a really cool way to do that. All right, so then we talked about cell culturing. We talked about DNA extraction. We talked about restriction enzymes, which cut DNAs in specific pieces. We talked about the fact we use restriction enzymes to create little places in plasmids for you to insert the gene you want, so you can do modifications of organisms. And we talked about how we use that in agriculture. We use that in animals. We use that to modify embryos of plants and animals. But we also use it in bacteria and yeast to make them factories for proteins that we want to do. There's multiple applications for medicine, for agriculture, that we use these kinds of things. We also use restriction enzymes to cut DNA into smaller fragments so that it's easier to sequence them, right? Which was something else I have to talk about. Now that you have multiple copies of the DNA, let's talk about shotgun sequencing. So shotgun sequencing. So then when we start a copy DNA, the idea was to copy the entire genome. But then Venture and his team came up with a different method of doing it, which was more efficient. So let's see if we find a good one here. So it's only about a minute. So let's see. It's probably all right.
Let us talk about the shotgun sequencing.
Oh, that's not easy,
The shotgun sequencing is introduced by Sanger et al. in 1977 for sequencing of genomes. But nowadays, the shotgun sequencing is modified in many tones, and we get many more advanced type of shotgun.
Sorry, it's not that I'm racist. It's just that since it's going to do a presentation, I can just do it myself. All right? So then, here's the idea. You have the original DNA, and this is why it's called shotgun. Because what does it look like you did with it? You blew it up, right? You shot it into tiny little fragments. And then you do the whole sequencing. By the way, it's the same idea of regular sequencing, except now you are sequencing all of them together and then using a computer device to assemble them into the order that they actually go into. And of course, you cut them with different restriction enzymes, right, to create different segments so that you can use the information from each segment to kind of overlap them, right? I kind of explained the logic behind this already, but how the sequencing period works. All right, by the way, this is showing you again how the computer overlaps them. So you have all these different segments, but there's information about how they match from other segments. And so when you look at one by themselves, you don't get the whole picture, but when you stack them up together, the computer does this, you can then figure out the actual sequence. It is actually faster to do it this way and have the computer reassemble it for you than do the whole thing from scratch one by one with regular sequencing. So sequencing relies on a method. You see these things? This is what shows up in a computer when you're sequencing a fragment. Right? Let me get closer. What it is it's showing you a peak. So it shows you that A peaked, and then A peaked again, and then G peaked, and then T peaked. Each color-coded here, it represents, there's a graph. And you see how in the background, all the colors are down here. But every time that color peaks, it means that base went at that point in the sequence. So when the device is reading the sequence, it reads the sequence of peaks to know the sequence of the bases. All right? But how does that actually work?
But how
It works like this. All right?
one bigger than
So basically, let's say you have to sequence that sequence, right? So you had to have all of these building blocks in the device. Are you with me? However, when a T gets used, there will be less T around. Does that make sense? And so the probe detects the drop in the T levels, if you think about it, right? A drop in the T level means the T is being used right now. Does that make sense? So if the T is being used right now, it means it was the one that came first. Does that make sense? Then another drop in T happens. That means another T went up. But then a drop in G happens. That means the G went next. Does that make sense? And so little by little, you actually build the actual sequence. But you have to do this one base at a time, right? And so, in other words, the first experiment you do, all you find out is that T comes first. The second experiment you do, all you find out is that T comes second. So you have to repeat this, the whole experiment, right, as many times as you have the length of the DNA. So you see, if you're trying to sequence something that's 1 million bases long, it will take forever. Because you have to do experiment once for the first base, again for the second base, again, are you with me? And so, it's smarter to do small segments and then piece them together than to try to do the whole thing. Does that make sense?
So why are some peaks bigger than the others? Like, is there more like in the other?
It's just how that reacts. I don't actually know enough about it to answer that question for you. But I'm sure in biochemistry you will learn that. All right? So, the point is, this is how we sequence DNA. Now, instead of just doing that for the entire genome, nowadays we break it into tiny pieces because it makes more sense. And that is what allows us to finish the genome, the first human genome. Instead of in 10 years, we finish in 2 years. And now we're sequencing everything we can. By the way, the first human genome was some person that got picked, I don't know who it was, to do it. Now, thousands of human genomes have been made. And you can sequence yours in less than 8 hours. Now, that's insane. We took a year to sequence the first one. And now you can do yours in 8 hours or so. Of course, you have to pay thousands of dollars to do it, but you can sequence your genome. And thousands of people's genomes have been sequenced by now. You know what I'm saying? And that's very interesting. Okay? So, that was biotechnology. We talked about, we never talked about electrophoresis, which is this. So then, you put your little DNA sample in the vial. Now, remember there's a current running through this, right? And one of them is positive, and the other one is negative, you see? Now, remember how DNA has those phosphate groups in it? Those phosphate groups are negative. Right? They have a lot of oxygens with negative charges on them. Because of that, they will be repelled by the negative anode and towards the positive cathode. So the current kind of pushes the DNA forward inside the gel. Now, of course, the gel is sitting in the bath of the electrolyte solution that's carrying the current. But the current is flowing this way, and the DNA flows with the current. The gel, however, provides a resistance that slows down the rate at which the DNA travels. Are you with me? However, larger pieces are going to be slowed down more by the resistance of the gel than smaller pieces will. Are you with me? And in that manner, this helps to separate DNA by size of the pieces. Now, of course, why are the pieces one side versus the other? That has everything to do with which restriction enzyme you cut. If you use this restriction enzyme, you're going to get 1, 2, 3, 4, 5 pieces. Right? 2 enormous ones and 3 smaller size ones. So then if you were doing the electrophoresis with that, you would see on the electrophoresis results you would have 2 enormous bands and then 3 little ones. Are you with me? The little ones go further because they have less resistance to the gel. But if you use the other one, you might have like 1, 2, 3, 4, 5, 6 small pieces all up there and no larger ones. Does that make sense? So the way the electrophoresis result looks, see here's an example, right? The way the electrophoresis result works depends on how you cut it. So let's say, for example, this is the starting point here where the negative anode was. This is the positive one. This is the direction of the flow right there. That means that this is a large DNA piece, right? And why? Let's say they were all the same sample, which kind of looks like it is. Why is it that this sample has lots of tiny pieces there? Because it was probably covered with a restriction enzyme, right? That broke into smaller segments. Now, if you work the same sample with a larger restriction enzyme, or the restriction enzyme that cuts into larger segments, then you get something more like this. Does that make sense? How do you use this? Well, remember, now I have it in pieces. But I wonder which of these pieces have the gene that I want. Then you do southern blotting on it, right? You put a bath that has the probes that you want, and then you can actually color code them to find the pieces that you want. One thing I want to show though is that how we actually use this. So, paternal testing, electrophoresis. And we're going to do the lab is all about this. The lab 9 is all about this. So we're going to pretend you're doing this in lab 9 basically, right? So let me see if I can do DNA. Okay. I'm going to get closer here. All right, so then if you have the mother, a child, and 2 possible males, right? Now, as you can see, the child does not have every band the mother has, because only half of what the mother has goes to the child. Notice I'm going to highlight some. This and this are shared with the mother. Does that make sense? You see this one and this one are shared with the mother. This one and this one is shared with the mother. That one and that one is shared with the mother. Then how could it possibly have these other bands? That's the father, right? So then you see this makes this father suspect. But it also makes this father suspect, so it didn't help me, right? But you see how this one is in this father, but not that father. All right, and this is in this one, and this, you see? And then this one and this one. So you see, that father is not really contributing. Does that make sense? So it looks like male 1 is the father. All right? You could also do something similar to this with crime scene. So if you're doing a crime scene profiling, all right? Oh, there's one here? Yeah. So you have the crime scene sample, the victim sample. Why is the victim there? Because you want to rule out the DNA as a victim's DNA, right? And then you have the suspects. Now you see this suspect's match pattern is the same as this one. Does that make sense? And so we think the DNA found the same is the DNA of the suspect, which is very different from the DNA of the victim, and therefore most likely the one that committed the crime. Does that make sense? So the idea of electrophoresis is one of the ways we use it is this way. But it's not the only way. We also use electrophoresis to separate things into fragments and then hybridize the fragments with probes to identify specific sequences that we're interested in sequencing. Then we go in there, grab the sequence, put it into the PCR machine, and sequence that, and find out the code. And then for that code, we do whatever we want to do with it. So that's some biotechnology stuff, and I hope you guys learned a lot about this. Now, if you look at the case, they actually, on 3A1, now we actually finished 3A finally. If you actually look at 3A1, it has some of these things we have to talk about. Electrophoresis, check. We're talking about plasmid-based transformation, yes, because we put, right? We talk about restriction enzyme analysis for DNA, yes. We talk about PCR, good. Genetically modified foods, yes. Transgenic animals and cloned animals, we talked about all this stuff already, all right? And pharmaceuticals, because remember we put the chemical that we want to create in the bacteria for them to make the drugs for us, because we don't have protein-making factories in the lab. They were called ribosomes. So we had to put them inside real things, right, to do that. All right, so then let's go back to what we were supposed to talk about today, which is or cell communication and gene regulation control. Now, we were starting to talk about this whole idea of DNA binding. That's how I got sidetracked to talk about the probes. Each of these activators has a domain that binds to a specific sequence of DNA, and because of that, they only activate specific genes, right? So that's what this whole idea of transcription factors. They will cause the fold that makes the RNA polymerase activate also. Now, so it's a combination of these control elements that makes each gene activate or not activate. And then also the idea then, how is a cell specialized? A cell is specialized because it has a lot of things. A specific DNA structure, fold versus not fold. A specific group of chromosomes in the areas that get read a lot. In other words, an analogy that was made with the library, it has certain books checked out and opened up in the table that's being read. Does that make sense? It also has specific lenses. I think of it this way. The book is own code. To read the code, you need a special lens. Are you with me? And those lenses are the specific transcription factors. So you're not going to read it unless you know how to read it. And you don't read it, it's a transcription factor. So that means two cells could have exactly the same folding pattern, and yet transcribe completely different genes because they have different chemical factors on. Different transcription factors. Are you with me? So you can have the same DNA and be different because of different folding. But you can also have the same DNA and be different because of different transcription factors which are there. But why are there different transcription factors there? Because of different signals that the cell is receiving. And we'll talk about what that more in next. Okay? Also, another thing that's important is that because the proteins are protein factors, results of genes themselves, the actual control mechanisms for a gene are all throughout the genome sometimes, right? And in fact, even the activator region sometimes is in a separate chromosome, which is yet another reason why chromosomes that are supposed to be on are all in the nucleosome region or regions where it is actually being activated. So nuclear architecture is very important. The DNA of each cell is the same, but if you look at each cell's DNA, it won't look the same. So if you could color code the DNA that's in each cell, every nucleus will have a different painting. Does that make sense? Because every nucleus will have a different set open, different sets in different regions, acting together to turn on, right? And so forth. So that's very important. All right? So the aggregate, and in fact, this is a question that Enzo asked that I never even mentioned. The cell goes as far enough to even, sometimes, let's say there's a book in the library that I don't want to read. All the pages are kind of, you got to fold this shit. But there's one page there that I need to do my job as a cell, whatever that job is. Sometimes what the cells would do is they would literally go to that chromosome, rip that gene out, cut it up, and bring it to the nucleosome region that needs to be read. Now, of course, if this cell is destined to be copied in mitosis, you're going to be able to go out and put it back there. If not, you're going to create a mutation, right? So this is more common in cells that don't undergo mitosis, like muscle cells, fat cells, and nerve cells. But still, every cell can do that. By the way, that piece of DNA that's out of place is also called a transposon, but it's not the same transposon meaning as the ones that tag along with the viruses between cells to become modified. All right? So with these mechanisms, cells can vary what they are transcribing. Sometimes, in fact, let's say, for example, there's a gene that I need to transcribe a lot. That means another thing I can do is, you know that page? I can make 10,000 copies of it and have it all over the place. That way, when the transcription factors are present, they will activate not one, but 10,000 copies. Are you with me? So sometimes, one of the things that cells do to specialize themselves is they make multiple copies of the same gene. Does that make sense? So that means that gene becomes overexpressed, and that's what makes that cell an expressor of that gene so much. So a cell that makes insulin probably has the gene for insulin copied multiple times in the nucleus. Does that make sense? So long as that cell is alive. So it's another technique that they use. This is the kind of thing we already talked about. But it doesn't end there. By the way, this is just showing you that whole idea of nucleosome, or a region of chromosomes that are working in one job. And sometimes they come from different chromosomes, right? But they all work together and activate each other. Either way, it's not over yet. Because the cool thing about all of this is that there are other levels. It depends which introns you remove. So that means it depends on which spliceosomes are on. It depends on whether or not the RNA actually makes its way all the way to the ribosome to be read. It depends on whether or not the protein, after read, actually gets packaged and folded the way it's supposed to be packaged and sent the way it's supposed to be sent in the Golgi apparatus. So it's not over yet. Even if you make the protein, you can still destroy it before it does its job, or fail to produce it correctly. That means, let's say the cell got a message, make some insulin, and now you have a bunch of RNA coming out of the nucleus, and all this is getting transcribed by ribosomes. And then halfway through the process, the cell gets another message from outside, say, fuck it, we don't need the insulin, cancel it. But all this insulin is already in the assembly line. You know what they do? They make chemical process to degrade all the insulin. So that means there's also post-translational control of expression. Remember, all the protein synthesis, all the chemical reactions, all the biochemical pathways are all enzymes, are all proteins. So everything you do is proteins. The structures, the functions, everything is done by proteins. That means your phenotype is protein-based, right? This is also part of 3A. But then that is happening because the DNA is on, transcribed, translated, and post-translationally modified. Which means yet another way that you can stop translation is by mess up with the folding, mess up with the processing, mess up with the delivery. There's a degraded protein even if it's already over. So the eukaryotic cell has many parts of the assembly line to stop production and prevent a phenotype to show up if necessary. Does that make sense? All right? And then along these lines, I want to just finish one thought about this, which is RNA interference. All right, so let's talk about that, which is very important. So then, small RNA control. This is called, there are different types of RNA. So then, looks like cell communication is not going to be yet today, but it's okay. All right, because I almost forgot about this. We already talked about many types of RNA, tRNA, RNA, mRNA. We also talked about SRP RNA, which was in the small recognition particle, which is a half protein, half RNA, recognizes the beginning sequence of a protein to grab it and take it to the ribosome if necessary. We talked about small nuclear RNA, which is part of the spliceosomes, to recognize the pieces that need to be cut on the introns. We talked about RNA that is part of the poly-A tail. We talked about RNA that's part of the telomerase. So all of these nuclear RNAs are around. But there's also some non-nuclear RNAs, which are very important. And this is very new biology, but the three ones that the book goes into and the EKs go into is miRNA, siRNA, and piRNA. Now, I already talked about some of them for you guys. The siRNA, for example, I talked about is RNA sequences which code for RNA, called siRNA, which their job is to recognize viral RNA. So you can think of them as RNA sequences which are complementary to viral DNA that the cell has faced in the past. So that means the cell has kind of like an anti-viral RNA recognizing molecule. So when this siRNA finds the viral RNA, it induces, all right, chromosomal folding. So in other words, if you find a prophage, remember viruses, the way they work is by inserting themselves in the genome to force the cell to make copies of themselves. But if you have a piece of RNA designed to recognize that as an invading piece, it will then act. So that, think of it, the way it works is that the siRNA is part of a larger protein complex. In fact, this is a whole motif of these RNAs. RNA works together with proteins a lot. So you have the siRNA inside of a larger protein structure. Now, when the siRNA recognizes the sequence, the larger protein structure does a job. In this example, the protein structure of which the siRNA is part of will actually induce, or will act as a repressor that will induce folding. That means, great, the viral RNA is there, but it's folded. So there's nothing that's not going to be expressed. Does that make sense? So it's a type of antiviral control mechanism, a kind of cell defense. piRNA is the opposite. It's the virus's anti-siRNA DNA. So that means some viruses have DNA designed to counteract the DNA that codes for siRNA. So I love it. It's kind of like biology warfare. So you have nuclear bombs, we have anti-nuclear bombs. Does that make sense? So the viruses have a mechanism to induce heterochromatin formation. Now what that is folding, right? True folding. So it means the viruses have this piRNA, which it will then go to your nucleus and fold the part that codes for the siRNA. So you say, oh, you have a secret agent to detect my virus, but before you recruit the secret agent, I would destroy the school that recruits it. Does that make sense? So now, this one is, we're discovering now in biology, is one of the most important types of RNA in the cell. Everybody learns about mRNA, but perhaps this is even more important. So the way the miRNA works, and I'm going to show you this, it's called RNA interference. I don't have enough time to show you animation and everything, but if those of you who are smart, Google it up, an animation of this. But I'll show you RNA interference. If you just Google RNAi, it will show up as videos. But I'm going to show you as an image and kind of talk about it really quick because I don't have a lot of time. But the way it works is this. So you have, find a better one that's simple. All right, this looks pretty good. All right, so let's say you have a piece of the cell's interior, right? And then you have a piece of RNA that needs to be transcribed by the ribosome, right? Well, in between, you can have a molecule, but actually this is actually showing you how the siRNA works. I want to show you the miRNA interference. They're all interferences of a kind, but I'm going to show the other one. Let me just type miRNA. miRNA. So, microRNA is what it's called. Find a good one here. Okay, here. So you have the messenger RNA just going through a ribosome to make a protein. But you have a microRNA that has a complementary sequence to a specific messenger RNA. All right? So you can think of this as a microRNA that recognizes that messenger RNA. Are you with me? So in between the ribosome and the nucleus, sometimes the miRNA will bind to the messenger RNA, recognize it, and then induce a chain of reactions. This is showing over here, right? They will induce a, so this is the miRNA piece, and that's the, they work together with proteins. So you can think of it as the mRNA is going to be carrying a protein squadron behind its back. I think of this as, if you watch Star Wars, the Clone Wars, there's always the clone troopers, but ahead of them there's always a Jedi leader. So I think of the mRNA as the Jedi general with the clone troopers behind him. So the proteins are like the clone troopers. They're the ones that are actually going to destroy the thing. But the mRNA is the one that's going over there recognizing what they need to do. So the mRNA has a complementary sequence which is going to target the messenger RNA in question, recognize it, and then the proteins will then work to destroy the messenger RNA. So what is this really? It's a mechanism to destroy a message that just came from the nucleus to make a protein. Why would you need it? Because you need an off switch. If the nucleus tells you, make insulin, and you don't have an off switch for insulin, you make insulin forever and maybe too much of it. So you need to have an off switch. And so mRNA is, we're thinking now, the off switch. So for every gene, there's a counter gene, right, of sorts. So there is these machines to destroy RNA to prevent a message from keep going. And so it has everything to do with gene expression control, because you stop the RNA from being expressed by destroying it before it gets to the ribosome, right? So that in order for a message to be expressed, it needs to be constantly be on. So the DNA needs to be on a lot. Hence why you need multiple copies of the DNA sometimes, so that multiple messenger RNAs leave, so that the policeman cannot deal with them, and then it gets expressed. Does that make sense? But then if the message of the nucleus stops, then these guys will little by little destroy all the messenger RNAs, and the proteins stop being made. So this is the off switch that we use to interfere with the message so that it doesn't continue forever. All right? And the siRNA is just a special type trying to interfere with the virus's message, if that makes any sense. Right? Okay, so that's gene expression control. So let me just make sure that we cover all everything the EK said to cover. So let's open them up and see if that's all right. So eukaryotes and bacteria both have mechanisms to regulate the expression of a gene to make sure whether or not something gets actually expressed. So I think we're going to go from this to cell communication. So that was good. 3B, gene regulation. And we did talk about all of this stuff here. Proteins that stimulate expression, we call transcription factors. So that was 3B1. Of course, we have to do 3C and 3D also.
And 4.
And so parts of 4. So we're quite behind. So the next thing to talk about is that the communication aspect of this. And so let's get to that. So there are 3 types of cell communication, right? But the most basic reason why you need communication is because cells need to know. Right, this is why we think they evolved. Why do a cell needs to talk to another cell? The whole reason for cell communication was sex. Because you need to know if the other person you're going to change genes with is of your kind. More importantly, of your kind of the opposite gender. Because here's the thing. Remember we talked about how meiosis evolved. So you have a situation where being 2 copies of the DNA requires more energy, more materials to maintain that status. So the cell has a mechanism called meiosis to reduce its gametes to, not as it is, its chromosomes to only 1 for times of hardship. But then you have to have a mechanism to go back to 2 for times of, because there's advantages for this. This provides more variety. This provides defense from a mutation because you have to have one bad, the other one's good. So diploidy is advantageous for a variety of reasons. So you want to have diploidy so long as you have the materials to keep making 2 copies. Of course, if you just double the DNA, so like say doing an S phase, without doing division, you don't really go back to variety. You just go back to 2 identical copies of what you had over here. So then sex comes in the picture here, so that another cell binds with this cell, so that you can have variety. So meiosis and mitosis are derivatives to just deal with this. Sex has to do with increasing variety on the process of going back up here. Now, to do this, you want to make sure that the one you're crossing with is not you. Not another identical copy of you. Because this guy here, before deciding to go back up there, could have done some binary fission, and now you have two of him. Right? And so later you want to go back up there with sex, and you just have sex with him, well, that might as well have done an S phase. Hold on, maybe I've gone fast there, so let me explain again. You want to have different copies, right? You want to have the DNA be varied. So sex evolved to make sure that's the case. So that means once I have the X here, I'm going to go back to this. So what do? Oh, I need a different color. So, hold on. So then, you want to go back up there. So what you do is you want to have sex with someone that's different. So when you go up there, you become varied. Correct? But when I was in the bad environment situation where I reduced my DNA with meiosis, early meiosis, right? Which was not about sex, it was about safety, about survival, right? Now, I could have just done the S phase. During the S phase without cell division, I go back to this. I duplicate my DNA. But that lowers the variety, so hence, let's do sex. But the thing about sex is that while I was like this, I may have done binary fission, right? And end up with two of me. So then in the attempt to have sex, I may actually have just found another one of me, if you think of what I'm saying, out there in the culture. And if I have sex with another one of me to go back up there, I go back without variety up there. Therefore, I might as well have done the S phase over here. And this whole thing was a waste of effort, the conjugation here. Right? And so, they got to make sure that this and this are the same species, but different lines of the same species. And there it goes, male and female. So this is male, and that's female. Only then they cross. Because then the female is different, by definition, because it comes from a different line. So male and female are not so much about having a different gender, it's about being a different version of the same type of life. So that you can make sure that when you restore unity, you restore with variety. Does that make sense? But for that to happen, these things gotta talk to each other. Be like, are you a yeast? Yes. Oh, good. But are you a yeast that comes from the same kind that I come? Ooh, sorry. No, I don't do that. You know what I'm saying? So, by the way, you could then argue, right, why is homosexualism a thing? Well, then you have to remember that that's much more complex, much later for other reasons. Definitely, though, gender is better for sex, obviously, right? But, of course, male and females, the way we see in humans, is a much more complicated version of two yeast talking to each other to make sure they're not the same kind. Are you with me? Or the same yeast species, but not the same line of yeast. Are you with me? So then you have on yeast, what you have is a membrane protein that is male and female. So that in one of them, you have a gap, and the other one, you have a phallus. Right? And then they only have sex with each other if they are opposites that can connect to each other. Does that make sense? And so that prevents them from having sex with themselves or with lines of themselves. Because the yeast can also just bud. Are you with me? Just split. So how does yeast make sure, when having sex, that it's not having sex with just a copy of itself? Right? By making sure it crosses with someone of the opposite type. Right? Does that make sense? So that is the origin of communication. The need to tell someone else, I am not you. Or not a clone of you. Does that make sense? Now there are three types of communication. There's local communication, and there's long distance communication. All right? And then there's direct cell-to-cell communication. So for example, if you tell a muscle cell to contract, that muscle cell will contract, but also several cells around it will also contract. How does that happen? It happens because between cells, there are gaps. All right? Like this, between them, called gap junctions in the cell membranes, where the chemistry of one cell can directly alter the chemistry of another. So that's called direct cell-to-cell interaction. Are you with me? Another example of this is in plants. They have something called plasmodesmata. They have literally gaps on the cell walls, just like the gap junctions, all right, which allow transfer of information from one cell to the other. So that means the chemistry of that cell, not directly, it's just a little gate, but it's kind of like the U.S.-Mexico border. There's Mexico and then there's U.S. But interactions between the two countries will influence both. Does that make sense? They're both independent unities. But because they're connected, they affect each other. Does that make sense? So all the marijuana going back and forth and the money going back and forth. Does it affect our culture? Of course it does. And so, does that make sense? So, the same way, that's direct cell to cell. Then there's local cell communication. So, the neuron that tells the muscle cell to contract is doing so through a synapse, right? Where it's throwing chemicals at the other one, and then at the other end there's receptors, right? So this one bypasses receptors because you don't need to go through the membrane. It just goes like through a little tunnel. Does that make sense? But when you're trying to tell one cell to the other how to communicate, that one you're going to have to produce chemicals here, which this cell then receives. So that means that cell has to have receptors for that. Does that make sense? But that's called local communication. Here's another example of local communication. When a white blood cell activates another one. So like in a white blood cell, we're going to talk about this time permitting. Right before the test, we're going to get to that part that talks about all the intricacies of the, because it has to do with cell communication. So it's going to feed off of this once we finish this, how the white blood cells talk to each other and activate each other. So that involves sometimes a cell touching the other cell and activating the other cell through receptors in the membrane. Right? But that's local. So whatever cell is over there gets activated. But then there's also long distance communication. Oh, another example there is cytokines. So you produce a bit of chemicals that call other white blood cells to come to their region. But to sense that chemical, they have to walk by their region. And it's local. So that's what's called inflammation, because one white blood cell that saw the invader produces the cytokines, and then all the other white blood cells nearby flock to from where cytokines are rare to where cytokines are common. And this is called taxis. It's a type of behavior where animals go towards a signal, right? So each white blood cell is following a taxis towards the region where there's more of that chemical, if that makes sense, right? And so because of that, it gets bloated with lots of white blood cells and water, and you get inflamed. So that's a local communication signal. However, there's also long distance. When your pituitary gland tells your adrenal gland to do something, or when your adrenal gland tells the rest of your body to do something, or when your ovaries tells the rest of your body to grow boobs, or when your testosterone asks everywhere. Those are long distance. How the hell is my kidneys talking to my brain? Does that make sense? That goes to the bloodstream. But what all of these types of communication have in common is that they have to start at the membrane where that signal has to be received. And I think of that as that, think about medieval times when you have this wall, although it's not a wall, it's a membrane, don't confuse with the cell wall. All right? Cell wall doesn't do this, does other jobs of maintaining structure. But you have in the membrane, you have these guys that are watching what's happening outside. And then when you receive a message, they will yell out to the inside what's happening outside. So since certain things have trouble going through the membrane, which is important because the cell needs to maintain homeostasis, but the cell also needs messages from the outside to go inside, you need to have this reception happen. Does that make sense? Then, once inside, the message gets to get to the nucleus. But the nucleus is far. And so what happens is a ping pong of sorts. When one cell tells the other, that tells the other, that tells the other, that tells the other, that tells the other, and eventually, not one cell, one chemical. So it's like, I activate you, which activates, it's kind of like a ping pong. So, but you haven't played that game? Of like, what happens with that is that telephone.
Oh.
Now, the cell's telephone, which is called transduction, which is the second stage of cell communication, is a little more complex than a telephone game. Because if we were playing cell transduction telephone game, halfway in between, one could choose to tell two. And those two could choose to tell two. And that message will go what? Faster and spread exponentially. Does that make sense? We also could have things like this going on, where the message is coming and someone says, nope, stop the message. All right? Also, there are certain situations where the message can become amplified, such that the person doesn't actually just even tell one person, but he yells it out. So this analogy is true for transduction as well. There's a lot of things that can happen in transduction with signal amplification, signal reduction, or of the message that's being passed from the membrane to the nucleus. Usually, this is called cascade, by the way. It's a cascade of chemical reactions that activates the final response. Now, the response is the final guy. that gets on receives a message. It's like, really? I'm on? I've been activated? So then what happens with that? By the way, this is how terrorism works, by the way. Alright, it really is. They don't tell messages directly to the cells. One cell tells another cell, and tells another cell, and they all activate. Does that make sense? So it makes sense because it's quieter that way. But either way, when the final guy gets the message, I gotta do it. I have now become activated. Usually, there's three types of responses. There's immediate response, where once activated, there's a protein that does a specific job, and it will get the job done. Let's say, for example, cyclins. So the end of the activation could just be a cyclin that gets turned on, and what is the cyclin going to do? Go activate a CDK, and now the cell's going to replicate. Does that make sense? Or, sometimes the activation, the final activator, is a transcription factor. So now the transcription factor is on. What does that mean? Some sort of DNA is going to be replicated. Does that make sense? So, here is what makes cells different from each other, though. Every cell has different receptors. So it means the receptors in the cell are different, which means each cell can only listen to certain messages. Are you with me? Every cell has a different network of transduction pathways. The chemistry of every cell, or the messengers that go from the net, are different. And then each cell has a different set of transcription factors on it. Are you with me? So each cell responds differently because it receives different messages, passes them differently, and does different responses. So that means even if two cells have the same receptors and the same transduction pathway, if at the end they have two different transcription factors, the response is different. Does that make sense? But if the cell has the same receptors and different messengers on the middle, the response is also different. So there is a million different ways to make a cell, depending on which receptors you have on, which messages you have on, and which transcription factors you have on. Now, why are those things that I'm talking about are all proteins? Why are they there? Because specific DNA was activated when the cell was going to cell school, becoming determined. So when the cell went to cell school, it activates different genes, which make sets of receptors, sets of messengers, and sets of transcription factors to be around. Which means that cell can only do certain things because it can only understand and activate certain messages. Are you with me? We'll pick it up from here and talk about how that actually works next class. The labs are on halt until otherwise. The calendar is insignificant. Alright? Just trust what I say in class until we catch up, which means we are working now, alright, on 3B and 3C. So that's what you should be working on reviewing. Also, more importantly, review step 3, the third 12th of your parts. Another set of FRQs this week, right? And so forth. Keep working on that. We will